Welcome, everybody. There are lots of names that I don't recognize, so really great to have you. I hope you join our webinars more regularly, and then some names that I do recognize. Wonderful to have you back as well. I'm just going to give everybody about a minute to log on, and then we'll formally start. And AI has joined us too. Welcome. Okay. Coming in automatically, which is wonderful. So I'm going to begin. Welcome, everybody. I'm Nicola Elliott-Wong from the H3D Foundation. And it's my great pleasure to host these webinars. We're halfway through our second semester program. And today we're very honored to have Rolf Müller join us to speak on the topic of natural products research. So Rolf Müller studied pharmacy and earned his PhD in Bonn, followed by research roles in Seattle and Braunschweig. Since 2009, he has been founding and scientific director of the Helmholtz Institute for Pharmaceutical Research Saarland, where he heads the Microbial Natural Products Department. He is also professor of Pharmaceutical Biotechnology at Saarland University. Rolf Müller is involved in several national and international research networks and collaborations, aiming to identify novel bioactive natural products, to analyze their biosynthesis and structures, and to develop their biological and chemical properties towards novel preclinical candidates. He is an elected member of acatech and Leopoldina, and an honorary professor at Shandong University, China, and at the University of Cape Town, South Africa. He was awarded the Gottfried Wilhelm Leibniz Prize in 2021 and the Charles Thom Award in 2023. We're so honored to have you, Rolf. Thank you so much. And without any further ado, but just to mention, we'll take a Q&A at the end. I'll hand over to you. Thank you, Rolf.
Yeah, thanks so much, Nicola, and welcome everybody to this webinar. For me, it's always a big pleasure coming back to Cape Town. Usually I like it better to be there in person, but the webinar gets us the opportunity to involve some more people. So as Nicola already pointed out, we are enjoying collaborations with UCT and also H3D for, I think, a decade now. So that is a lot of fun. So let me start out with what I would try to entertain you with in the next 45 minutes. So this is about natural products research and but also some general approaches towards novel anti-infectives from microbes. So we usually focus on microbes. You all know about the global burden that antimicrobial resistance puts on us. There's a Lancet paper that shows approximately 1.3 million deaths per year are directly associated with antimicrobial resistance, with additional 5 million which are indirectly caused by antimicrobial resistance, and this is known for a while, so this is nothing new. It's also on the political agenda. We clearly need new antibiotics. So antimicrobial resistance has a lot of issues. The use of antibiotics, in agriculture, the misuse of antibiotics. I cannot cover all this. For this, you would need a whole lecture series. What I will be talking about is the lack of antibiotics in the field and what we try to do about it using natural products. So when it comes to antimicrobial resistance, traditionally, there's always been the question, what is the most important pathogens on a global scale? And as you can imagine, each and every medicinal microbiologist, depending on their country of origin, would give you a different answer. And luckily, a couple of years ago, WHO did us a great favor and declared a list of priority pathogens in critical group, high group, medium group, where obviously MTB is a very important part of, but it's largely gram-negative pathogens that cause a lot of difficulties in the hospitals, and that can, depending on the geographic region where you're from and the healthcare system that you are at, can have very different interpretations. But we tend to look at this on the global scale and hope to work on those pathogens which or where new antibiotics would have the highest impact. Now, many people ask me at this stage already, why are you doing this? Why isn't this something that pharmaceutical industry should do? My answer to that always is pharmaceutical industry indeed should do it with the help of academia. However, and this is again a very short introduction into a big mess that we call global market failure in this area. Currently, you don't make money by generating antibiotics, and pharma generates a lot more revenue in cancer, respiratory treatments, cardiology, etc. In fact, the last few companies that developed new antibiotics even went bankrupt. So, and that is a big issue in itself, that we all together have to make sure that our societies value antibiotics the way they need to be valued, because otherwise we will not get pharmaceutical companies back into the game, which currently is a big issue. Again, that's something you could talk about for an hour. It's a fact. We need to deal with it, and that causes even more pressure on academic science to come up with solutions. The other question you get, what is actually in the pipeline? And if you look at the phase 1, 2, 3 compounds which are currently in the clinical pipeline, the total numbers, they on first sight don't look too bad. So this is 159 according to the recent analyses of the pipeline. However, if you now look at each and every compound and ask the question, is this a me too, meant to get some of the market share, or is it really an innovation which you can use to treat multi-resistant strains? Then things look very different. So for example, if you only take new chemical classes, you end up with just 10 compounds that are left in the pipeline. And if you now reduce it to those that exhibit a new mode of action, you're left with 3. So very little. And you then ask the question, what are they intended to be good for? They all target MRSA. So basically, currently, there is no single compound in clinical phase 1 to 3 that would have a novel chemical scaffold and a new mode of action against gram-negatives. And that is quite daunting and clearly shows we need new antibiotics here. And we need more of you to join these efforts. We in our institution are at the Helmholtz Society in Germany. Helmholtz is one of the larger extra-university research organizations in Germany, like Max Planck and Helmholtz and Fraunhofer and Leibniz. We have a number of fields we work in, and in the research field health, there's one center for infection research, which is located in the north of Germany. For those of you who know the German map, or who don't know the German map, I put it in here. Germany is very small, so I think it can fit into South Africa 5 or 6 times, according to my knowledge. This is where Braunschweig is located, here in the north. Here's Berlin. And we are located here in the south, in Saarbrücken, in a small state called Saarland, which is right at the French border. We call it the German West Coast. And the center has 3 major research topics. One is on pathogens, one is on immune response and interventions, and then there is the third major topic, which is anti-infectives. And within anti-infectives, the site in Saarbrücken plays the major role with a lot of PIs. I show you all of the PIs in this space, located in Braunschweig at HZI and at the HIPS. This is the team. You probably know some of them if you are from the natural products field. We have hired a few new PIs in the recent past. Then we have a strong team in MedChem. We invested quite a bit in bio and chemoinformatics in the last couple of years, and we also do drug delivery. So this is the general setting of our institute. So for today, I will zoom in into natural products, where we have a strong expertise. For those of you who are not that familiar with natural products, they are, in terms of their structures, a bit more diverse than the typical chemical libraries. Their three-dimensionality is given, and they have this enormous advantage of having been evolutionary optimized as molecules. However, they obviously have not been evolutionary optimized to be used in humans. So they are optimized for their targets and for whatever was the use for the producing organism. And we often have to deal with modifications that make them applicable for humans, or we deal with toxicities that make them not applicable in humans. Here's a number of examples from the literature. You know penicillin, you know Taxol. There's epathalon, which is an anti-cancer compound. And if you do the statistics, almost all antibiotics that are currently in use are of natural origin or derivatives of these compounds. And we continue in this field because we still believe there's a lot to discover, and the approach involves all sorts of modern technologies which are applied in this field. And I'll try to show some examples. Now, first of all, you would ask the question, where do they come from? The microbial sources are largely actinomyces and streptomyces, which have traditionally been used in pharmaceutical industry over 50 or 60 years. We have a number of fungi that produce natural products, Pseudomonas, Bacillus. And then there are some upcoming producer classes which have much less been studied, which gets you the advantage that the chances of finding something new are higher. Nevertheless, the technologies available for actinomycetes because of the long-term studies are much better. So we work with both. We work with streptomyces, actinomyces, but our favorite organisms are the myxobacteria, from which quite a number of natural products have already been isolated. I will introduce this class of bacteria because you certainly sit on them. They are ubiquitous. They can be isolated everywhere, and they are quite fascinating. So if you've not heard of them, I hope that I can convince you that they are the coolest microorganisms on the globe. So they do all sorts of things you do not expect from microorganisms. They can swarm, they can glide over an even surface. Once they starve, they form what we call fruiting bodies. So this time-lapse movie shows you the formation of a fruiting body on a piece of bark. These fruiting bodies are up to 1 millimeter in size. They build within 2 or 3 days. And within the fruiting body, spores ripen that await improved nutrient conditions, so then a swarm can form again. And the swarms and the various growth stages of these bacteria produce a lot of secondary metabolites. And this is what makes them so interesting to us. Here's a few examples of compounds we've isolated with various biological activities. Now, maybe with respect to producing antibiotics, the most fascinating feature of myxobacteria is that they are predators. So if you like, they are something like the Tyrannosaurus rex of the bacteria, and they are on top of the food chain. They would basically sit everywhere in almost every soil sample you see, and here is a quick time lapse that shows you what they do. What you see here is single-cell Serratia species on an agar plate, and you now have to imagine the myxobacteria on the upper left. These are rod-shaped cells, so you can see the first ones coming in, and then they sense their prey and they realize, wow, this is a big buffet here, but I need my friends to consume all of this. And then they move back to their swarm and tell their friends where the buffet is, and together they then can produce a lot of exoenzymes and also secondary metabolites, which they use to consume their prey. Yeah, and at the end of this time lapse, you can see that, this has not had a good outcome for Serratia, but Myxo has found a lot of energy to digest. And I can assure you this is happening in basically every salt sample you can take worldwide. And these bacteria are heavily understudied because of difficulties isolating them. So what we basically do in the lab is shown on this slide as a summary. We do go for worldwide sampling, all according to Nagoya protocols. We then culture these bacteria. We search for bioactivities in the extracts, and if we find something promising, we would try to isolate the compound that is behind this activity, do its structure. If it's novel and promising, we would want to know how does it actually work in mode of action studies. And if this is still promising, we would try to produce more for further studies into animal studies and also engineer or chemically synthesize the compound so we can ideally go into an application. So obviously, this workflow starts with a lot of samples and ends with very few, because most of the compounds are not amenable to all what I'm telling you here, or they're not useful because of toxicities, etc. But we always look for those that we think we can develop further. And towards the end of the talk, I will give you a few examples where compounds have come out of this process and are now getting close to clinical application. I should say that microbiota as a source have become quite important to us with the natural product knowledge. We are now also studying the correlation between human disease and the constituents of microbiota with the aim to understand which secondary metabolites play a role there. So this is becoming a major focus in the lab. If you want to help us with isolating Myxo, we have generated a citizen science project, so please check out our webpage. We are happy to send out sample kits. There's also an app that tells us where exactly the sample was taken, and then we would also be in touch with you if something interesting comes out of the sample. We are now also involved in metagenomic analyses of these samples to prioritize what we can get out of them. That's the start. Once you have an isolate, it looks like this. But when I started my research, it would have been a dream to get the genome sequence of just one of these. Nowadays, we sequence them first and then look for their metabolites. And we've recently finished a study where we sequenced 130 myxobacteria from various resources and analyzed these in great depth to understand what their potential for secondary metabolite formation is. So the genetics now come first because sequencing has become so easy. And what is very important also in comparison to other bacteria, that whatever has been isolated and grown in the lab is very little in comparison to what is out there in nature. So for example, what you see here is before this study, which is currently being written up, there were descriptions for 10 family of myxobacteria and 31 genera of myxobacteria. And with this study, we will bring this to 94 genera and 28 families. To put this into some perspective, if you work in the actinomyces field and you find a new genus, this is already almost a sensation. With myxobacteria, we find them on a quarterly basis. And that is very exciting because the more diverse the bacteria are, the more diverse their secondary metabolism is. We then look into the genomes and look at what we call biosynthetic gene clusters. So that is basically areas on the chromosome where they have all genes required to make a specific metabolite. And these gene clusters, each of these gene clusters is basically in charge for making one compound class. And as you can see here, previously we had 460 of these biosynthetic gene clusters in the public domain. MeBig is a reference library where well-annotated biosynthetic gene clusters are located. And based on the study that I'm just describing, we will have more than 2,000 biosynthetic gene cluster types in the future. So this is a heavy transformation of what we have in the myxobacteria. Now people will ask, how does this compare to actinomyces? Of course, actinomyces, there's hundreds of groups that have been working on this for decades, but still, in the actinomyces, there's currently 25K gene cluster families. And with this study in Myxo, we add roughly 10%. And these 10% we add with just 3% of the strains. So these 25K gene clusters came out of 5,000, approximately 5,000 sequenced strains in the actinomyces. We only had to sequence 130 strains and come up with 10% of the gene cluster families, which is amazing. But even more importantly, this overlap here is very little, which tells you it's very unlikely that a myxobacterium would make the same compound as an actinomycete. So in other words, we are likely to find novel chemistry. So this is just genes. We of course want compounds. I show you one example of one of these families, which we called Pendulispora. We originally called it Streptomyxo because the appearance of the strain on agar plates was this whitish, like actinomycetes spores, but then we saw that the strain started moving, so it was a myxo, and it's a new family. Some of them isolated from Zambia, by the way, some from the Philippines. And we then started looking at their biosynthetic gene clusters with the genomes again. Basically, they are all new. And there's a very recent paper here that came out in Chem this year where we've shown the first couple of completely novel secondary metabolites including such which show decent activity against gram-positives and fungal pathogens. This made it to the cover of Chem if you're interested in this paper. So in general, if you look at metabolomes of myxobacteria, we could correlate in a study where we looked at 2,500 metabolomes, we could clearly correlate their phylogenetic distance with the production of different metabolites. Now we have all the genomes, so this makes the analysis easier, but at the end of the day, this is what the whole field sees, independent of the class of bacteria. These bacteria are full of these BGCs, which are shown here in green and red. But if we grow these bacteria under standard laboratory conditions, they would only express few of them and all of the others would be silent. This is why I put some in green here and others in red. And one big miracle in the field, how do we get the products of all these silent gene clusters? Some of the approaches involve change of the growth conditions, like you would just move myxo from liquid culture to solid culture and have them move around. They already start making different compounds. Well, what has turned out much more successful in our lab is co-cultivation technologies, where we basically put them on the same agar plate with either another myxo, with a fungus, with Pseudomonas, and then have them do their thing. And we extract and see how production of metabolites changes, and it significantly changes. This is what I can tell you from these analyses. We do this in assays as described here. So we put myxo on a plate, have the compounds diffuse, and then use the diffusible compounds to do a second step MIC assay. Or we put a pathogen on a plate and myxo together with it, and again check the diffusing compounds in a MIC assay, and then compare this with what we see produced by myxo when it's just grown by itself. And from these analyses, we get a lot of new bioactivities. However, because we're only doing this on an agar plate, it's difficult to upscale. So the production scale here is a clearly limiting parameter, which we have to deal with. And therefore, we came up with some technology, which is now in its third phase, so to speak. So basically, idea was, why don't we have larger plates and then pump the media over a absorber resin to bind the compounds generated and then move it back to the plate. We first had a steel version of this, which was a bit complicated. Then we had a 3D printed version of it, which never survived the autoclaving process. Now we have a form in which we can basically print these plates and use them for higher productivity of these samples. And from this, we currently have 3 strains that produce something which is new to us and has anti-gram negative activity. And hopefully I can show structures according to this assessment in one of my future talks. Now, all of these workflows end up in MUXO extracts, which we are happy to share. There's extract libraries. There's new compounds from them. We are currently generating an extract library also with fractionated extracts. And you know, for people who are interested in screening them, please make contact. We are happy to share this and then see whether, depending on the bioactivity testing you are interested in, we would find something new from MUXO. So this is clearly different chemistry than what we see in the field. So this is all set up in an automated fashion. The libraries are stored. We have a liquid handling workstation, and the screening plates can be made according to your needs. We can do this in 380 rail format. So this all depends on your setting if people are interested in collaborating with us on screening the MUXO chemical potential. Now, all this work leads to quite a number of papers, which is fascinating. But then for the next step, we asked ourselves, what are the most important projects to us? And as we are at an infection research center, we came up with a list of what we want these projects to fulfill. And everything we call a promising candidate profile would be a compound that addresses clinical need. I've explained what that means by the WHO priority list. We would want it to have new chemistry, so we do not want to work on stuff that has been described in the past and where there's little chemical modification possible. Ideally, we would want it to have an innovative target, so there's no cross-resistance. And last but not least, we wanted to show proof of concept in animal models. So these are, I would say this is the bar raised quite high. And most of the projects we have at this stage cannot proceed. But luckily, we have a couple of promising candidates which would fulfill all these needs. And here is the list of compound classes that we currently have for which this is all true. New chemistry, activity against WHO priority pathogens, proof of concept in usually mouse models of infection, or even a dog, sometimes even monkey models. And then we have a new mode of action for all of them. So I will not have the time to guide you through all of this. Cystobactamid is a new broad-spec anti-gram negative for MUXO. There's a lot in the literature if you want to read about it. Coramycin is another completely novel compound class where we have not yet completely figured out the mode of action, but it's new. Also broad-spec gram-negative against for MUXO. We work on the chlorotonyl species, which is broad-spec anti-gram positive and also highly promising anti-malarial. For all of these, there is recent publications. If you want to hear about them, and for the folks in Cape Town, they know about our joint work in the DNA end targeting griselimycins, myrcoplanecins, which we currently follow up in the TBDA. Now, for the rest of the talk, I will focus on these 2 examples, this corallopyranin and darobactin. And let me first introduce corallopyranin to you. This is an antiparasitic agent. It shows very good activity against filariasis, which is a devastating disease in Africa. It's a worm disease, so it's a bit counterintuitive to use an antibiotic. The reason for this is that the worm harbor a endosymbiont called Wolbachia. And it turns out once you kill the worm, the immune system can kick in and clear the infections. So we have teamed up with Achim Hörauf's lab and other collaborators at Bonn University within the German Center Infection Research. Achim's team, they are the experts on this worm disease. And they used corallopyronin in animal models, and they could show that the adult worm burden, as shown here in a dose-dependent fashion, is heavily decreased if you use this molecule. The molecule itself, we studied for a while. These pyrone antibiotics, they are RNA polymerase inhibitors targeting the switch region. So like rifamycin also being RNA polymerase inhibitor, but with a different binding site on RNAP, giving the explanation why there is no cross resistance. And we have studied how this compound is made. I will spare you the details. It's quite a complicated biosynthesis with many steps, where a red and a blue polyketide chain is generated, and then a miracle enzyme comes in, which we studied in vitro, that then condenses the two chains to form the pyrone. Now, why is this important? That information here on the gene level is one of these biosynthetic gene clusters I mentioned earlier, and it's roughly 55 kilo base pairs, and it harbors all the information required to make corallopyronin. And this turned out to be essential for the project because the original producing strain, the Corallococcus, had a very low production yield, and the production was not really reproducible. Some people say it produced only according to the moon phase, and therefore the project would have been stopped without an alternative to produce the compound, as there was no total synthesis, and there still is no total synthesis available. And the idea we came up with here was, why don't we transplant the whole biosynthetic gene cluster into a host that we can genetically manipulate and that is close to Corallococcus. And we took our pet host Myxococcus xanthus, and the overall idea is shown here. You simply take the genes that are in charge of making these compounds, and then you design them either in vitro, or you synthesize the genes, you reassemble them, put them into the heterologous host, and ideally you get your compound made. You can also do this with unknown BGCs to discover novel chemistry. This is one of the major topics in the lab. And for corallopyronin, this turned out and worked like a charm. As shown here, we could deregulate the gene cluster, put it into M. xanthus. We have optimized yields here now. The strain moved to our production facility in Mark Stadler's lab in Braunschweig, where multi-gram production was feasible. And then the compound could go into preclinical development, where also formulation was improved heavily by Karl Wagner's lab in Bonn. All the ADMET study looked safe, so this made us very optimistic about the project. The oral bioavailability is improved. We got formal advice from the BfArM, which is the German regulatory authority for this, and we also got some additional funding to then produce this compound on the 15 cubic meter scale in the bio-based production facility in Belgium, which is required for the closing, or hopefully closing, preclinical studies. And for this, we managed to get funding together with Eisai company earlier this year, and the GHIT fund in Japan luckily agreed to fund this project for the next maybe 1.5 years, when it is scheduled to go into phase 1. So this involves some finalizing in vivo toxicity studies, and yeah, keep fingers crossed this can make it into phase 1/2A. Now here's my second example. I will try to move you through this quickly. This is a compound class discovered by the Lewis lab in the US, darobactin. Quite fascinating, not from Myxo, but from insect pathogenic bacteria. And we knew from older studies that gene clusters from insect pathogenic bacteria we can well express heterologously. And these insect pathogenic bacteria turned out to be difficult as production hosts. However, the Lewis lab and their collaborators in structural biology, the Hiller lab in Basel, they have shown that the darobactins address a very attractive target in the outer membrane of gram-negative, the BAM complex. And this is an essential complex, and when you inhibit it, you get killing of gram-negatives. So this caused us to get very interested in this molecule. We used our heterologous expression technology to design biosynthetic gene clusters in silico that would then generate modified derivatives of the compound. And we've by now made, I think, almost 100 modified derivatives of the compound. And with the structural biology knowledge, we could also selectively increase and improve bioactivity. Here's one example for Acinetobacter. You can see this is darobactin A, the original compound. This is D9, which was one of our first front runners, and now we've come to D22, which shows significantly improved activities on Acinetobacter and also some other of the important species. This was corroborated by structural biology studies where cryo-EM gave us the information how the derivatives would bind and how binding affinity can be improved. And then we went into a second round of optimization, and nowadays we work on more chemical derivatizations by semi-synthesis of the compound class. We then moved the compound class into initial zebrafish models where they showed nice activities. Here is pharmacokinetics. You can see high metabolic stability and plasma stability, which then encouraged the first in vivo studies. And I'll show two in vivo studies for the end of this talk. Here's a serious infection, peritonitis in bacteria. And with darobactin, we could reach 100% survival in this, which is very significant. And here, meropenem was used as a comparator drug. The compound's also very active in urinary tract infections. So we also ran some urinary tract infection models in mice. These are shown here. So here the dose reduction and dose dependency of reduction is also very significant. So we are very hopeful with this compound class that it can be developed further. In vivo proof of concept is given now, and the whole program aims at increasing the in vitro activity, improve further the safety profile or prove the safety profile, which so far didn't show red flags. And one big issue is always the production and the cost of goods. This is what we are trying to address. So let me summarize. I hope that I could give you an impression of why we study myxobacteria, although we do not neglect actinomycetes. All this work led to 6 natural product classes, which have novel chemistry, new mode of action, no cross-resistance, and in vivo proof of concept. Of course, there's a long way to go to get these into application, but we try with many collaborators to do our best. We've seen the examples of darobactins or corallopyranins, where genetic heterologous expression platforms turned out to be essential for the development of these compounds. And all of this would not have been possible with a lot of talented and motivated people. So this is my own group. But I should say I mentioned a lot of collaborators, especially the late stage projects are always done in larger consortia. Otherwise, we couldn't put this together. I appreciate also our collaborations with UCT. Maybe some people from Digby's or Kelly's or Val's lab are in the audience. Thank you so much for listening. I'll be happy to take questions.
Rolf, thank you so much. Everybody, I have enabled you to unmute yourself, so please raise your hand if you have a question. You can also write it in the chat if you like. And I'm just going to give people a minute to think about it. But I think from my side, Rolf, when did you become aware of natural product research as a field that you wanted to give your professional life to? I know we have a lot of emerging scientists in the audience who are interested in, how do you get to where you are?
I think there's two questions in one, right? Getting aware of this. I studied pharmacy. So as a pharmacist, you get aware of natural products and their role pretty early in your studies. A decision to devote your professional life to this can typically come later. I think, during my PhD, I got fascinated by this, but then moving into a professional academic career, there's opportunities and either you take them or you leave them. I also, for a while, thought I would end up in industry. But then as a postdoc in the US, I realized I really am having fun with this and also some success. And then there were opportunities in Germany, which made me feel like for myself, working with young people and being involved in education is much more fun for me than just staying in industry. So I've never regretted this decision.
I think an international component to anybody's career is vital in this field. Are there any opportunities in the with the institutions that you work at for mentorship or things like that, especially for our Pan-African scientists? Is there any way that
Of
they can
course.
engage?
Of course. I mean, we currently have, I think Gottfried is here in Anna's lab from UCT. So we've had several people from UCT working with us. I'm also happy to engage further in like the welcome workshop that took place in UCT a couple of years ago, and we are happy to engage in joint PhD studies or postdocs. We have the collaboration with Val and team, and also Digby in the TBDA. I think there's always opportunities, and we love to do this. My team, I think it's 50% non-Germans. And the Germans, a lot of them is the technicians who typically don't move that much. So if you want to apply, don't worry about the language. The lab language is not German, so it's also not French. So that's all done in English.
Thank you. Okay, are there any questions? I'm not seeing any hands raised. Okay. And please don't unmute if you aren't going to ask a question. All right, I can hear a few voices. So here's a message for you, Rolf. Thank you very much, Prof. This is quite amazing. I've always been interested in natural product research. Only have one question. Is it hard to get a good yield of these phytochemicals? If so, why is it hard?
It depends. The production scale is really very different depending on the organism. So sometimes this is the main limiting factor. Sometimes you find an organism that easily produces hundreds of milligrams. So there's no general rule. So luckily, we now have all the genetic means to change, for example, the expression of the genes, which is often the limiting factor for the production. You also need to understand how the compounds are made. So sometimes the building blocks are limiting, then feeding can resolve these issues. And at the end of the day, sometimes it's total synthesis, right? If the molecule is not too difficult to make, we don't care, right? I want to develop compounds, whether it's at the end of the day made by fermentation or by total synthesis. We want the best of both worlds. Quite often we also combine it. A short question, long answer. There is no general answer for it. So because you need to think about why is the organism producing the compound, right? And in many cases, we just don't know. And under which circumstances is the organism producing the compound? So if we knew this, it would be much easier to understand also how to make more. And sometimes, especially if you work in the antibiotic field, you have to think about this. This is gram-negative bacteria, and we are looking for anti-gram-negative compounds. So, that poses a question. How do they become self-resistant? Aren't they limiting themselves by producing these compounds which are toxic to their competitors? And then we look at self-resistance mechanisms and how they are expressed and how can they protect the cells. And using overexpression of self-resistance mechanisms, you can also induce production. So there's various ways, and every case is different.
Bauer, go ahead. I think you're next in line.
Thank you, Nicola. That was a wonderful presentation. I have a question with the compounds that you were able to isolate in larger quantities. Did you guys do some biotransformation studies to get some analogs for structure-activity relationships?
That is something we do. Typically, the bacteria produce a family of compounds because the biosynthetic proteins are not that specific. And the first SAR studies you do by isolating all the congeners, right? And then you already have an idea. Next idea we have is changing the genes, as I've shown for the darobactins, to deliberately change the structure, which in some cases works very well. Then chemical semi-synthesis is used, and we will also use some enzymes to modify compounds, as you suggest. So biotransformation is taken into account, but it's not our first bet. It also depends on how much we know about the target. If we have a co-structure of target with a compound bound, so whether we just need compounds to see what we get, or whether we can deliberately change areas on the molecules because of the knowledge of the target compound binding.
Okay, thank you. I have another second question. I'm not sure if you understand it, but I would like to ask it. It's a crazy question. So say hypothetically there's this compound that has only been isolated from plants. It has never been isolated from microorganisms.
Mm-hmm.
Is it possible if, say, maybe you take your streptomyces or your myzobacteria and you find a similar BGC, if I can call it that? Like now, how possible is it to bioengineer the microorganism into producing a compound that it has never produced before, that you are specifically targeting a class?
Oh, that's possible. I mean, I've shown examples, right? So the darobactin, E. coli never made darobactin before, and we made it make it by using the genes from another microorganism. Also, the corallopyranine, we moved the gene cluster from one species to another. I'm not saying this is easy, but it has worked in many cases. When it comes to making plant products in bacteria, this is a field in itself, but there's also successful examples where people, for example, produce Taxol in bacteria or in yeast. There's quite a number of compounds from plants that were already made in bacteria, which involves a lot of engineering usually. It's doable. And in some cases, there has even been traditional knowledge or papers that claimed compounds are made by plants, but eventually it was shown that it's not the plants themselves, but
it's the endophytes.
Bacteria from the rhizosphere or fungi from the rhizosphere or the endophytes making it. And there's even cases where the endophytes make something which the plant then modifies a bit. So this is a fascinating field.
Nice, awesome.
Haruna, you were next if you'd still like to ask your question. Although you did lower your hand. Okay, let's move on. And then the next question, what is your go-to method for cloning large gene clusters?
That's a so for the bacteria, what we use the ReadyT technology, the recombinering technology, with gene synthesis becoming less expensive. We sometimes also synthesize up to 5 kb fragments and then use yeast tar cloning to assemble them. And for those of you who want to know the most recent technology, keep an eye open on Science. There's a paper coming out where we have data on a new method to clone large gene clusters.
On that note, I have put all the links for both Rolf and H2D Foundation, as well as a feedback form if you'd like to offer feedback on the webinar in the chat. There are a few more questions if you're up to it, Rolf. One is, did you use natural products from medicinal plants?
Actually, this is how I started my PhD, trying to work out biosynthesis of medicinal plants products. But at that time, at that time, genome sequencing of bacteria was not possible. Genome sequencing of plants was completely out of range. Nowadays, sequencing plants is possible. So I think this adds a lot of potential to plant natural products. My lab has focused on bacterial natural products. We've had a few people who came, I think mostly from African countries, with the idea of indigenous plants and identifying compounds. So we've done this in the past, but it's not a focus in our lab.
Thank you. Okay, next one. Can you please share with us the process and problems encountered with the patent filing of these natural products and their semi-synthetic derivatives?
Oh,
Patents.
That's a question for a patent lawyer, and it even depends on the country where you do this. For example, in the United States, since maybe a decade or so, you cannot patent a natural product anymore. But you can patent a new synthetic substance. So which to me doesn't make any sense, because if you were to find a natural product and you claim you synthetically made it, you can patent it. Anyway, so the patenting is always, it's a weird process, and the patent laws are, out of my know-how. Of course, we need to make sure we at least have freedom to operate. So if we have a new natural product, the way to patent it in our case, and also on the international side, is that you patent a new natural product or natural product for a application. This is feasible. Just patenting the chemical matter is basically impossible. So what you can patent is a cystobactamid or darobactin for treating Acinetobacter infection. This is possible. But how exactly this is done, I can give you examples, but what you exactly have to look at and how you write a patent it's an art in itself. It's completely different from writing papers. You put in examples which are only done in theory. So for the scientists, I think this is a tricky process, and you need experts to help you with it. Otherwise, you're certainly not safe.
Thank you. And one last question we'll take. Do you make use of computational methods in natural product discovery? Which softwares are used in your research center?
Yeah, I indicated in the beginning, maybe I can, is my slide presentation still on? Can you still see? No? Anyway,
no, sorry,
it doesn't matter. In the last 4 years, we've hired 4 bioinformaticians and chemoinformaticians, because, this is really a field without which I don't see any future. If you just look at the genomes I presented, 130 genomes times 15 megabase pairs. I forgot to mention Myxo has the largest genomes. Analyzing these without specific bioinformatics expertise is not feasible anymore. We, of course, use software like anti-smash, you name it, but these guys in our lab, they developed their own software. Quite a bit of it is already on the web pages, and luckily, we can convince them to produce the tools and the technology that is required for our mass spec analyses, for our dereplication, and also for the genomic and transcriptomic analysis. Otherwise, we would be lost. You know, we can generate that many data that it's impossible to evaluate. And there's so many data types that there's not a single software to be used. For the genome analysis, probably anti-SMASH is the most widely used tool. But we've, for example, come up with a tool how we analyze PGCs from microbiota, which got published last year. And there's specific tools depending on what you want to do. A lot of these efforts go into dereplication based on mass spec. So you directly have a pipeline from your extracts to the high-res MS mass spec data into databases like GNPS or MSLib, where all the natural products and their data are found. So you know as quickly as possible if what you see as a bioactive is a known compound or something that is new. And we want to know this as early as possible in the process so we don't run after compounds which have been isolated and elucidated already.
Rolf, thank you so much. I know there are still more questions pending. I think a lot of them have to do with, you know, are there opportunities for internships and things like that, which I think you've already answered. And I've put the links up in the chat. So everybody, please do reach out to the various institutes that Rolf is connected to see what opportunities are available and follow them online. Social media will also publicize those opportunities. So with that, I'm going to close the session and just to say a huge thank you, Prof. Müller, for giving us your time and your expertise. You're a very busy person and we really appreciate your input on our series. So thank you so much.
Yeah, thanks so much for listening. And there's always opportunities, right? Germany has the German Academic Exchange. Check out their webpage at daad.de. And there's Humboldt Foundation. So, you know, if you really want to move and get the experience, get in touch, you'll find a way. And the best of luck for your research career.
Thank you. And thank you to all our listeners and all our participants. You've got my contact details, so reach out if they have more follow-up questions and I'll try and direct them. Thanks everybody so much. See you next time. Bye.
Bye-bye.
Bye, thank you.